Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing West Bengal set question paper for the subject Life Science. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of West Bengal set exam, you can go to my channel Omni Bioentrance. In the channel select playlist, in the playlist select WB set and there you will find. So coming to the first question. In Arabidopsis, apicobasal polarity during embryogenesis is established by which of the following genes? So it is GNOM, that is option C. It is a member of the ARF-GEF family, that is ADP ribosylation factor, guanine nucleotide exchange factor, and it lead to establishment of apicobasal polarity. Whereas FACEKEL, FACEL, it is involved in embryonic patterning and auxin distribution. Shoot root, it is involved in root development and differentiation, and Gurke, it is involved in ovule development. Then colchicine is a chemical which, so colchicine is a chemical which prevents microtubular polymerization. Answer is option A. And it inhibits microtubule formation by binding to tubulin protein, which are the subunits of microtubule. And this prevents polymerization of microtubules. And you all know these microtubules, they are involved in spindle formation, and this leads to cell cycle arrest during the metaphase and thereby preventing cell division. So prevents microtubular depolymerization. It is not depolymerization, actually it prevents polymerization. So B is not the correct answer. Activates attachment of spindle fibers with kinetochore plates. So colchicine, it disrupts spindle formation rather than promoting attachment. So that's also not correct. Prevents polyploidization of cell. Colchicine, it actually promotes polyploidy by preventing cell division. So option D is also not the correct answer. Identify the event which is not associated with Na plus current impairment. Exposure of actually Na positive current impairment, it refers to disruption to the normal flow of sodium ions into a cell, and this is typically through the help of voltage gated sodium channels. So Na positive current impairment, it can result from various factors like toxins or diseases or medication or even certain kinds of trauma. Okay. Then so here exposure to red tide, so they have asked the question as which is not associated with Na positive current impairment. So here exposure to red tide caused by dinoflagellates. Yes, red tide can produce brevetoxins which can block sodium channel. So it is associated with Na plus current impairment. They have asked not associated with. So A is not the correct answer. Consumption of shellfish fed on dinoflagellates. Yes, shellfish, they can also accumulate brevetoxins which can cause Na positive current impairment in humans who consume this shellfish. Then consumption of cadmium contaminated water. Cadmium is a heavy metal, and it can also have various toxic effect. but it is not associated with Na plus current impairment. So that is the correct answer. They have asked which is not associated with Na plus current impairment. Consumption of ovaries of bowlfish. Yes. Bowlfish ovaries, they contain tetrodotoxin and it also blocks or impairs Na plus current impairment. Then next one. Cortisol released from adrenal cortex in response to stress lead to. So it leads to mobilization of free fatty acids. So cortisol mobilize energy stores to help the body respond to stress. So cortisol, it stimulates the breakdown of triglycerides in the adipose tissues and thereby release the free fatty acid into the bloodstream. Then how many neurons are likely to be there in your brain? Answer is option C, 15 followed by 9 zeros. A single nucleotide polymorphism is an example of. So single nucleotide polymorphism, it is a genetic marker. Single nucleotide polymorphism, it occurs when a single nucleotide, that is either your A, T, G or C, it is altered at a specific position in the genome. And they can be used as genetic markers because they are inherited in a Mendelian fashion and they are abundant in the genome, and they can also be found associated with certain traits or diseases. Transpositional control, it is the control of gene expression by transposable elements. Genetic regulation, that is control of gene expression. Then a frame shift mutation, it is a type of mutation that occurs when a nucleotide is inserted or deleted, and it can lead to a change in the reading frame. So if you know the other three, that is A, C, D options, then happily you can go for B. Then which of the following assumptions does not belong to Hardy-Weinberg principle? So according to Hardy-Weinberg principle, there should be a large population size. The gene pool, it remains constant. Then there is random mating, there is no mutation, there is no gene flow, that is there is no immigration or emigration. Then there is no natural selection, and the allele frequencies, they remain fixed through generations. That is the assumptions of Hardy-Weinberg equilibrium. So they have asked here which does not belong to Hardy-Weinberg principle. So gene pool remain constant, that is as per your Hardy-Weinberg equilibrium. Then uses algebraic equation, yes, they have used algebraic equations. Allele frequency varies in a species, that is not correct. Allele frequencies, they remain fixed as per Hardy-Weinberg principle. Frequency remain fixed through generation, that is correct. That is as per your Hardy-Weinberg equilibrium. The type of male sterility where pollen development is normal but pollen function is impaired is termed as. So pollen development is normal but pollen function is impaired. This means that pollen grains are produced normally, but they are unable to fertilize the egg cell, leading to male sterility. That is pollen development is normal but pollen function is impaired. So that type of male sterility is called as functional male sterility. Answer is option B. Sporogenous male sterility means occurs when there is a failure in the formation of spores, leading to abnormal pollen development. Structural male sterility means there are physical or structural defects in the anthers or other pollen or floral organs, leading to abnormal pollen development. Then transgenic male sterility, it is through genetic engineering, that is induced through genetic engineering process, where you introduce a gene to disrupt pollen development and function. Then next one. In which minor phyla of invertebrates the anus is located inside the lophophore? So it is in the case of Foronida. Answer is option D. The next one. Under stressful condition, the administration of non-steroidal anti-inflammatory agent, that is NSAID, such as aspirin, may cause significant reduction in. So, aspirin inhibits the production of prostaglandins, and these prostaglandins, they help to dilate renal blood vessels, increasing blood flow to the kidneys, and increase glomerular filtration rate or GFR, and helps in proper waste removal. When these non-steroidal anti-inflammatory agents such as aspirin inhibit prostaglandin, then renal blood flow and GFR will decrease and leading to reduced kidney function. So the answer here is option A, that is GFR. Then which of the following is not a G protein coupled receptor? So out of these, it is glycine receptor. It is not a G protein coupled receptor. They have asked which is not a G protein coupled receptor, whereas the others are adrenergic receptors, that is the one which respond to epinephrine and norepinephrine, that is a G protein coupled receptor. Glutamate and muscarinic receptor are all G protein coupled receptors. The basic factors that stimulate gastric secretion, that is gastrin, it is a hormone released by the G cells of the stomach lining, and acetylcholine, it is a neurotransmitter, and histamine, it is a chemical released by enterochromaffin like cells in the stomach lining. So these are the basic factors that stimulate gastric secretion, that is gastrin, acetylcholine and histamine. Then secretin and cholecytokinin, they stimulate pancreatic secretion, not gastric secretion. Okay, then somatostatin, it is a hormone that inhibits gastric secretion rather than stimulate it. And vasoactive intestinal peptide, it relaxes the smooth muscles and inhibit gastric secretion. So they are not the correct answer. Then which of the following cellular enzyme is related to relaxation of vascular smooth muscle, that is ENOS, that is endothelial nitric oxide synthase. See the other options. You could have, if you know the other three options, that is B, C and D, then you could have happily marked A. So hexokinase, you know, it is involved in glycolysis, and it is used for the conversion of glucose to glucose 6-phosphate. Catalase, that break down hydrogen peroxide into water and oxygen. Then phosphofructokinase, again involved in glycolysis, catalyzing the conversion of fructose 6-phosphate to fructose 1,6-biphosphate. So if you see the B, C, D option, you can happily go for option A as your answer. Which of the following physiological functions is most likely to be impaired due to the presence of mutated occultin and claudin proteins? So occultin and claudin proteins are found in the tight junctions, and these are specialized structures that prevent the free passage, or they create a barrier against the free passage of molecules, and they are usually found in the blood-brain barrier cells. So what they have asked, which of the following physiological function is most likely to be impaired due to the presence of mutated occultin and claudin proteins? So mutation in occultin and claudin proteins can disrupt the formation and function of tight junctions, and they can impair blood-brain barrier function. So the answer here is proper functioning of blood-brain barrier. Then conduction of electrical impulse through intercalated discs of cardiac tissue. So that is mediated by gap junctions, and gap junctions are formed of connexin proteins and not occludin and claudin. So that makes A as the wrong answer. Then C, if you see generation of coordinated peristaltic waves in the smooth muscle cells of gastrointestinal tract, that is also not related with occludin and claudin proteins. Then proper assembly of gap junctions again by connexin proteins, not occludin and claudin. The mid-blastular transition is the point in the development when transcription of zygotic genes begin. Answer is option C. Activation of 1 minute. Translation of maternal mRNA is initiated. So that is during the oogenesis and early embryogenesis stage, not during mid-blastular transition. Cell division in the embryo ends. It does not end in the mid-blastular transition, but it continues with some changes. Then blastocele formation occurs. It occurs during the blastula stage and it is not a characteristic of mid-blastula transition. Then next one. In Drosophila, bicod gene controls embryonic gene expression by transcriptional activation and translational repression. If BCD mRNA is injected at the middle of a bicod mutant Drosophila embryo, what would be the expected phenotype? So first I will tell you the BCD gene, it is involved in the anterior posterior axis formation during embryogenesis. Okay. And BCD mRNA, it is localized in the anterior pole of the egg, and it is involved in the head and thorax development. So if BCD mRNA, it is injected into the middle of a bicod mutant Drosophila embryo, the injected mRNA will be translated into BCD protein and form a gradient along the anterior posterior axis. Now the BCD protein gradient, it will activate expression of the genes that are involved in the head and the thorax development, and its location will be in the middle of the embryo. So as a result, the embryo will develop head-like structure in the middle and telson-like structure will be present at both the termini. So the answer here is formation of telson at both the termini and head at the middle. Answer is option C. The next one. Which one of the following redox centers with the respiratory chain does not possess a prosthetic group? So flavoproteins, that is proteins in with that is your FMN and FAD, and they have flavin as the prosthetic group. So they have asked not possess a prosthetic group. Iron sulfur proteins, again they contain iron sulfur clusters as prosthetic group. Ubiquinone, it is also known as coenzyme Q, and it is a non-protein component of the electron transport chain, and it does not possess a prosthetic group. So answer here is option C. Cytochromes, they contain heme as prosthetic groups. Then why do Bruxelles zebras migrate 150 miles every year away from Botswana's water-filled Okavango Delta to the Makgadikgadi salt pan? So this is to lick the salt there and absorb the mineral it provides. So that is the answer, and the minerals they provide for zebras health and well-being, and it is good for the lactating female and the young foals of zebras. Okay. Then next one. Plasmodium species overcome the host physical and physiological barriers by synthesizing the protein named. So it is called as thrombospondin related anonymous protein or TRAP. Answer is option B. So the TRAP, it is a surface protein and it is expressed by the Plasmodium sporocytes, and this help the sporocyte to adhere to cells such as hepatocytes and erythrocytes, and it help in the parasite migration through the host tissue and also evade the host immune response. Then next one. A 24-year-old foreign male PhD student of your university was detected with blood in his urine after 4 weeks of his arrival from Africa. So one of the student is working along with you, and he is been detected of urine of blood in the urine after 4 weeks of his arrival from Africa, and the microscopic examination of the urine revealed the presence of eggs with terminal spines. So presence of eggs with terminal spines, that is one point you have to remember. Then detected blood in urine after arrival from Africa, that is another point. In course of the investigation, he admitted that he had been working in the rice field of his family occasionally since his teenage. The most likely etiologic agent of his complaint was, so he has been working in the rice field in Africa, and in the rice field of Africa, schistosoma hematobium is very common. Okay, so he must be the etiologic agent for his complaint will be schistosoma hematobium. Based on all these points, that is detected blood in the urine after coming from Africa, then its egg urine revealed the presence of eggs with terminal spines. Schistosoma mansoni, it is not for urinary schistosomiasis, it is involved in intestinal schistosomiasis. Then Ascaris lumbricoides, it is for intestinal infection, it is a roundworm. Then Trypanosoma brucei, it is a parasite that causes sleeping sickness, not urinary tract infection. So that's also not the right answer. Rocky Mountain spotted fever is transmitted by Rocky Mountain wood tick. Answer is option D. Then the inheritance of a trait due to difference in DNA methylation pattern can be referred to as, it is called as epiallelic inheritance. And this DNA methylation, it is a type of epigenetic modification, and it can affect gene expression, and there will not be any effect in the DNA sequence. So it is epiallelic inheritance. So genomic inheritance, it is due to difference in the DNA sequence of the genome. Mitochondrial inheritance due to difference in the DNA sequence of mitochondrial genome. Then oligoglycosylceramides present ubiquitously in plasma membrane that play a very important role in signal transduction, cell-cell interaction, and modulation of cell proliferation is a class of, so they are a class of glycosphingolipids. Glycosphingolipids, they have the ceramide backbone. Okay. And they are involved in signal transduction, cell-cell interaction, and in cell proliferation. This one, glycoglycerolipids, they contain glycerol as backbone. So that cannot be the answer. It is oligoglycosylceramide. So it should have ceramide as their backbone. So it goes for glycosphingolipids. Then glycoprotein means carbohydrate chain attached to the polypeptide backbone. Then phosphoglycerides, they are lipids that contain glycerol backbone linked to phosphate groups and fatty acid chains. Then next one, the ABO blood type of 1000 people from an isolated city were determined and the following data are obtained. So A type is found in 42 people, B in 672, AB in 36 and O in 250. What would be the frequency of I allele of ABO blood group gene from these data? So the small I allele, it corresponds to the O blood type. Okay, so we have to find the frequency of the O blood type. So the calculate the I frequency represent the O blood type in ABO blood group use Hardy-Weinberg equilibrium. The ABO blood group has three alleles, that is IA for A blood type, IB for B blood type and small I for the O blood type. P plus Q plus R is equal to 1. So P refers to frequency of IA, Q refers to frequency of IB and R refers to that of small I. So P plus Q plus R the whole square. Here there are three alleles that are involved. So we have to do P plus Q plus R the whole square. So that is expansion will give you P square plus Q square plus R square is equal to 2PQ plus 2PR plus 2QR. So frequency of A allele, that is capital A plus A O will be P square plus 2PR. Frequency of B allele will be Q square plus 2QR and frequency of AB allele will be 2PQ and frequency of O allele will be R square. So as per the question they have asked frequency of O allele, so you have to find that of R square. So frequency of O allele is R square. So 250 divided by 1000. Since there are 250 people in the O blood type as per your question. Okay, that will be 0.25. So that will be 0.5 will be the answer. Then next one. In a food web consisting of 10 species, the observed number of links is 20. The connectance value of this food web is. So connectance value of a food web is given by L by S square, where L is the number of links in the food web and S is the total number of species in the food web. So substitute the values and you will get it as 0.2. So the answer is option A. Then next one, a keystone species is, so it is any plant or animal species that plays a unique and crucial role in the way an ecosystem functions. That is keystone species. Then next one. Which of the following is not the characteristic of mangrove vegetation? So, for viviparous germination, yes, mangrove seeds, they germinate while still attached to the parent plant. Then pneumatophores, yes, they have specialized roots that grow upward above the water level to obtain oxygen. Because mangroves are, they have low oxygen environment. Then prop root, that is which are like stilt roots that provide additional support and stability to the plant. Okay, so the option which is not the characteristic of mangrove vegetation is buttresses. They are not found in mangroves. They are typically found in the tropical rainforest trees. Then raceways are typically used for the culture of. So it is used for the culture of trouts. That is option D. The next one, several techniques are employed for the aquaculture of Indian major carps. Which one of the following technique is not recommended for this purpose? It is inbreeding, because inbreeding it can lead to loss of genetic diversity, it can reduce fertility, and it can increase the susceptibility to diseases, because inbreeding is breeding that between closely related individuals, and this can result in the expression of deleterious genes. So inbreeding is not recommended for this purpose. Induced breeding is that you use hormones to stimulate breeding in the carps. Then selective breeding, it is a selection of individuals with desirable traits, such as those with fast growth. That is selective breeding. Then polyculture is culture of multiple species together with Indian major carps. So this can increase the overall productivity and diversity of the aquaculture system. Then next one. Which of the following statement is not true? Viruses contain either DNA or RNA as the genetic material. That is correct. The nucleic acid of viruses is surrounded by a protein coat. Again correct. Viruses multiply within living cells. Yes. Using the viral mRNA and tRNA and ribosome. That is not correct. They do not use their own mRNA and tRNA, but instead they will use or they will hijack the host's mRNA, tRNA and ribosome to replicate their own genetic material. That is what the viruses do. So that is the answer they have asked, which is not true. Viruses multiply inside the living cells. That is also correct. Then next one. In some organisms, the potent allosteric activator of phosphofructokinase 1, one of the key regulatory enzymes of the glycolytic pathway. They have asked for the allosteric activator of phosphofructokinase 1. So it is fructose 2,6-bisphosphate is the answer. So for fructose 2,6-biphosphate, it is produced by the enzyme phosphofructokinase 2, and that is the fructose 2,6-biphosphate. It increases the activity of the phosphofructokinase 1 in glycolysis. Okay. Then ATP, it is an allosteric inhibitor, not an activator of phosphofructokinase 1. Then lactic acid is a product of anaerobic glycolysis, but not an allosteric activator of phosphofructokinase 1. And phosphoenol pyruvate, it is an intermediate of glycolysis, but it is not an allosteric activator of phosphofructokinase 1. Then anaerobic ammonium oxidation, ANAMOX, is an important microbial process involved in the nitrogen cycle. Select the correct options from among the following combinations. The ANAMOX bacteria oxidize ammonium with nitrite under anaerobic condition. That's correct. The ANAMOX process is currently used for the removal of ammonium in wastewater system. That is also correct. The ANAMOX bacteria belong to genera Brocadia, Cunea, Scalindua, Anamoxoglobus and Jettina. That is also correct. So P, Q, R are correct. That makes option B as the answer. But as per me, S is also correct. The ANAMOX bacteria are characterized by the possession of extracellular anamoxosome, in which ANAMOX process takes place. That is also correct. but they have not given in the options, but they are given PQR, so that is correct. Then next one, an increase in entropy occurs in which of the following situation? Capturing energy from sunlight during photosynthesis, that involves the conversion of light energy into chemical energy, and there is decrease in the entropy. Then consolidation of memory of dendritic spines, this involves the reorganization of neural connection, again decrease in entropy. Then active transport of Ca2 plus from cytosol to endoplasmic reticulum, it also uses the energy to move ions against their concentration gradient, and there is decrease in entropy. Release of inorganic phosphate during ATP hydrolysis, that is the correct answer, because during ATP hydrolysis energy is released as phosphate groups and resulting in the production of ADP and PI. So it increases entropy of the system. So answer is option D. Then next one, which of the following groups of microorganism produce the neurotoxin beta-N methylamino L-alanine, BMAA, a natural non-proteinogenic diamino acid that is produced in marine, freshwater and terrestrial environment? So it is cyanobacteria that is producing this neurotoxin. Then next one, biosynthesis of which of the following pairs of antibiotics is achieved through the use of non-ribosomal protein synthesis, NRPSS. So it is thyrosidine and gramicidines. Answer is option A. Then next one, the leader peptide of TRP operon contains 2 contiguous tryptophan residues. Yes. So the leader peptide of the TRP operon, it is 14 amino acid peptide, and it has 2 contiguous tryptophan residues. In absence of tryptophan ribosome, stalling occurs, allowing the leader region 2 and 3 to pair. At what position of the 14 amino acid leader peptide do contiguous tryptophan codons located? So your real question is, at what position of the leader peptide are the 2 contiguous tryptophan codons located? So these tryptophan codons, they are located at positions 8 and 9 of the 14 amino acid leader peptide. Then next one, siRNA forms a complex in spliceosome. So before that, siRNA, it is involved in RNA interference. Okay, that is RNAi, and it helps in the regulation of gene expression. So siRNA, it is complementary to specific mRNA molecules, and they form complex with the RISC, that is RISC, that is RNA induced silencing complex protein, and they will bind to the complementary mRNA molecule, and it will inhibit the translation of the mRNA or cause its degradation. So siRNA forms a complex in spliceosome. Spliceosome, it is involved in splicing of the pre-mRNA molecule, and siRNA is not involved there. Then siRNA recruits histone acetyl transferases to the nucleus. It does not do so. It is not involved there. Forms a complex with RISC protein to inhibit translation or cause degradation of complementary mRNA. That is correct. Then is not transmitted to daughter cells after cell division. That is also correct. That is, it is not transmitted in the way your DNA is transmitted. But for your information, I am telling you that siRNA can be inherited through a process called as RNA induced inheritance, where siRNA is transmitted to daughter cells and the mechanism is entirely different. Okay. Then a phenomenon where a single gene has more than one phenotypic effect, it is called as the pleiotropy. That is option A. Then what is this phenocopy? It is a it is a condition where the environmental factor can cause a change in the phenotype of the organism, and this change, it will mimic the effects of a genetic mutation. That is phenocopy. Then phenology, it is the study of events such as flowering, migration, breeding in relation to various environmental factors, and phenotype you all know. A decrease in BCL2 Bax ratio will impair a cell by. So BCL2 and Bax, they are proteins that help in regulation of apoptosis. So the BCL2 or Bax ratio, they have asked. A decrease in the BCL2 Bax ratio. So BCL2, it is an anti-apoptotic protein and it inhibits apoptosis by preventing release of the cytochrome C from mitochondria. That is what BCL2 does. It is a protein that inhibits apoptosis. Whereas Bax, it promotes apoptosis by inducing the release of cytochrome C from mitochondria. So when BCL2 Bax ratio decreases, it means that there has to be an increase in the Bax level relative to BCL2. That is, when the denominator is increasing and the numerator is decreasing, only then there will be a decrease in this ratio. Okay, that is what it means. Okay, so Bax is increasing means Bax is the one that is promoting apoptosis. So apoptosis will be promoted when there is a decrease in the BCL2 Bax ratio. So the answer here is option B, that is inducing apoptosis. Then next one. One of the underlying reasons for the development of tertiary lymphoid organs, that is TLOs, as lymphoid neoorganogenesis in case of cancer is. So tertiary lymphoid organs, they are non-lymphoid tissues in response to chronic inflammation, especially in the case of cancer. And they have asked reasons for the development of TLOs. Okay, so if you suppress TN alpha is a cytokinin that can promote the formation of TLO. So suppression of TN alpha will likely inhibit TLO formation. They have asked reasons for development, not for suppression. So A cannot be the answer. Then chronic inflammatory microenvironment. Yes, that is the answer. Because the inflammatory microenvironment in tumors can lead to recruitment of immune cells, that is your lymphocytes and dendritic cells, and they can lead to the formation of TLOs. Then inactivated IL-17 mediated immune response. So IL-17 again a cytokine, and it lead to the formation of TLOs. But inactivation of IL-17 would inhibit TLO formation. So that cannot be the answer. Blockade of IL-22, it is also a cytokine that can promote the formation of TLO. So blockade means inhibition of TLO formation. So that is also not the answer. While lambda DNA enters a new host, how is lysogeny established in the absence of C1 repressor? So the answer here is option D, that is with the help of product C2 and C3 necessary for RNA polymerase to initiate transcription at the promoter PRE. So PRE promoter, it is responsible for the expression of genes necessary for the establishment of lysogeny. Okay. Then lambda zap vector, it is an example of phage mid. So phage mid, it is a hybrid vector that combines the features of phages and plasmids. So answer is B. Then viral based transfection involves using a viral vector to carry a specific nucleic acid sequence into a host cell. Which of the following viruses are often used for stable transfection of cells? So answer is lentivirus. Then adenoviruses, they are used for transient transfection. That is for a short period of time. Then adeno-associated virus also same way. Herpes virus are not used for transfection application. So answer is option B. 3 restriction endonucleases PQR are capable of recognizing 468 base pair sequence respectively. What would be the relative frequency of occurrence of these sequences in the bacterial genome? So the frequency of occurrence of recognition sequences in the bacterial genome, it is inversely proportional to the length of the recognition sequence. That is, longer the sequence, the less likely it is to occur in the genome. So the one with the 4 base pair, that will have the highest frequency. That is, it is shorter sequence, so more the chance of occurrence in the genome. So P will be the highest. Then comes 6 base pair, which is having lower frequency than the 4 base pair, and 8 base pair, it is the lowest frequency and the least common because it is the longest sequence and less likely to occur by chance. So P greater than Q greater than R is the answer. Answer is option A. Then mini satellites or variable number of tandem repeats are used in DNA fingerprinting. Answer is option C. Then in gene therapy, VNTVRs are not used. Gene mapping also not used much. Then polymerase chain reaction, it is used to amplify specific DNA sequences. Not primarily PCR is not used for VNTR applications. It is in DNA fingerprinting that you use. Choose the right combination of components required to set up a PCR from the following. So it will be your template DNA, that is the DNA sequence that you want to amplify. Then you have the 2 primers, that is your forward and reverse primers. Then DNTPs, that are the building blocks for the DNA synthesis. DNA ligase is not used because it is to seal the gaps between DNA fragments, and PCR is involved in amplification of DNA sequence, not in joining of 2 DNA sequences. So DNA ligase in A and B, so it is not the right answer. C, they have given template RNA, which is again not correct. D, if you see, it is template DNA, 2 primers, DNTPs and DNA polymerase. Yes, DNA polymerase is the enzyme which is used for PCR amplification, that is your Taq polymerase. Then next one, in protoplast culture experiment, the early detection of primary cell wall formation can be achieved by. So protoplast, you know, they are cells without a cell wall, but upon culture they can eventually regenerate their cell wall. So by staining the protoplast with chalcofluor white, you can visualize the formation of the primary cell wall. So early formation of primary cell wall in protoplast culture can be detected by calcofluor white. Then next one. Which of the following embryo sacs comprise an egg apparatus of 3 haploid cells, 3 diploid antipodal cells and a central cell with 2 polar nuclei, of which one is haploid and the other is triploid? So this type of embryo sac, it is the polygonum type. Answer is option C. Then next one. In cryopreservation experiment, cryoprotectant chemicals act by. So cryoprotectants, they are chemicals that are usually added to protect the cells and tissues from damage caused by ice crystals. That is, which are formed during the freezing process. So these cryoprotectants, they lower the freezing temperature of the cytoplasmic fluid. Answer is option B. That is how they act, these cryoprotectants. Then image formation in a scanning electron microscope requires signals of. So when the primary electron beam, when it hits the sample, it will lead to the emission of secondary electrons. So these secondary electrons, they are detected by the detector and it creates the signal which is used to form the image. So image formation in scanning electron microscope requires signal of secondary electrons. Answer is option D. So that's it for now. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of West Bengal set exam being discussed, you can go to my channel Omni Bio Entrance. In the playlist, select for WB set and you will find them there. Thank you.